Scientific significance and comparison of a 16-year quantitative study on lifestyle, effort versus biomarker improvement. Number 1265, March 4, 2025. Gerald C. Shu, eclaireMD Foundation. Category: Obesity and Diabetes. Abstract. Introduction. In his previous research paper on the Overall Metabolism Index comparing the author's data with U.S. national data, as shown above, the author observed the following. The differences in 4 key biomarkers, body weight, glucose, blood pressure, and blood lipids, between the author and the U.S. national averages were not as large as the differences observed in 6 lifestyle details: food intake, hydration, sleep patterns, stress levels, exercise habits, and daily routines. In other words, the biomarker differences were smaller than the lifestyle differences. This unexpected observation sparked the author's curiosity and led to the investigation presented in this paper. This particular study, using the data collected during the past 15 years, from 2010 to 2024, the author conducted a longitudinal self-study, tracking the direct relationships between his 2 specific lifestyle interventions, and 2 corresponding biomarker improvements. Specifically, he quantified how reductions in his food portion size and carbohydrate and sugar intake, grams, affected changes in his body weight, measured each morning, and daily postprandial plasma glucose, ppg, levels. The key results from this study are as follows. A 74% reduction in food portion size led to a 25% reduction in body weight. An 81% reduction in carbohydrate and sugar intake led to a 28% reduction in ppg. Therefore, the calculated lifestyle effort to biomarker improvement ratios are approximately 3.0 for both weight and glucose improvements. In other words, it required about 3 times the lifestyle effort to achieve 1 time of biomarker improvement, when compared with the following 3 published clinical studies. In the diabetes prevention program, a 7% weight loss was associated with a 58% reduction in diabetes incidence, corresponding to a ratio of about 1 to 8.3, meaning the disease risk reduction was significantly larger than the lifestyle effort. In the look-ahead trial, an 8.6% weight loss led to a 0.6% reduction in hemoglobin A1c, with an effort to result ratio of approximately 14.3 to 1. In the Finnish diabetes prevention study, a 5% weight loss resulted in a 58% reduction in diabetes incidence, corresponding to a ratio of about 1 to 11.6. It is important to note that the author's personal longitudinal study differs fundamentally from these 3 large-scale trials. His study directly measured biomarker shifts, such as changes in body weight and glucose levels, in response to specific lifestyle interventions. In contrast, those 3 major trials primarily measured event risk reductions, such as the future incidence of diabetes, across general populations. Fundamentally, these 2 approaches address different objectives and outcomes. His data are individual, precisely quantified, and collected daily over 16 years, capturing true biological cause and effect relationships, unlike the group averaged and statistically inferred results of large studies. Therefore, the ratios from large population studies and this personal study are not fully comparable. Nevertheless, the findings are consistent with the broader scientific understanding that lifestyle efforts must significantly exceed the magnitude of biomarker or health improvements, defining the effort to result ratio as the lifestyle effort percentage divided by the biomarker improvement percentage. The author's study results indicate an effort to result ratio of approximately 3.0. In contrast, major population studies often show either very small lifestyle efforts, achieving large reductions in disease risk, or significantly higher effort requirements when focusing on direct biomarker changes. Detailed data summary. Weight and food, from 2010 to 2024. Body weight, BW or M1, dropped by 25%, from 220.00 pounds to 164.56 pounds. Food portion size, M9A, dropped by 74%, from 1.76 to 0.46 normalized units. 1.0 in it indicated 680 grams per meal. ppg and C and S, from 2014 to 2024. Postprandial glucose, ppg or M2, dropped by 28%, from 137.27 mg per deciliter to 99.36 mg per deciliter. Carbohydrates and sugar intake, C and S grams per meal, dropped by 81%, from 72.00 grams to 13.76 grams. These findings further confirm that it requires approximately 3 times greater effort in lifestyle improvements, specifically reducing food portion size and carbohydrate and sugar intake grams, to achieve a one-time improvement in body weight and glucose biomarkers. Pathophysiological explanations. 1. Body weight versus food portion size, M9A. Body weight is primarily determined by the balance between energy intake and energy expenditure. When food intake is reduced, the body initially loses weight. However, metabolic adaptation occurs, lowering basal metabolic rate (BMR), and making further weight loss increasingly difficult. Despite a 74% reduction in food portion size, only a 25% reduction in body weight was achieved. This demonstrates the body's natural homeostasis and survival mechanisms that resist excessive weight loss by slowing metabolism and increasing hunger hormone secretion, such as ghrelin. 2. PPG versus carbohydrates and sugar intake, C&S. Postprandial glucose levels are tightly regulated by insulin secretion from the pancreas. Reducing carbohydrate and sugar intake significantly reduces glucose spikes after meals. However, factors such as impaired insulin sensitivity and beta cell dysfunction influence glucose levels. Even small amounts of carbohydrates can cause large glucose spikes in individuals with insulin resistance or prediabetes. Despite an 81% reduction in carbohydrate and sugar intake, only a 28% reduction in PPG was achieved, indicating persistent cellular dysfunction, such as impaired glucose transporter 4 transporter activity and beta cell stress. 3. The 3 to 4 times effort principle. Both weight control and glucose control exhibit nonlinear responses to lifestyle improvements. Biological systems, including metabolism and glucose regulation, are highly resistant to change due to metabolic adaptation, hormonal feedback loops, and long-term cellular damage. Therefore, achieving biomarker improvements requires 3 to 4 times greater lifestyle effort than the magnitude of the biomarker shift itself. 4. Additional considerations. Adipose tissue produces inflammatory cytokines that impair insulin sensitivity, making glucose control more difficult even after weight loss. Long-term lipotoxicity, characterized by fat infiltration into the liver, muscles, and pancreas, further impairs metabolic and insulin secretion capacity. Consequently, substantial and sustained lifestyle interventions are necessary for meaningful improvements in body weight and glucose levels. In short, the author's observations are consistent with known human pathophysiology. The body fiercely protects its weight and glucose levels for evolutionary survival, making the reversal of chronic conditions such as obesity and type 2 diabetes disproportionately difficult, relative to the magnitude of effort required. Graphical evidence supporting the 3 to 4 times effort principle. The x-axis of the first diagram, left side, shows lifestyle improvement effort percentage, such as reductions in food portion size and sugar intake. The y-axis shows biomarker improvement percentage, such as reductions in body weight and PPG. The diagram illustrates that biomarker improvements increase nonlinearly with lifestyle efforts, indicating that greater efforts are needed as improvements progress. The second diagram, right side, displays 2 orange bars representing lifestyle efforts, reductions in food portion size and carbohydrate and sugar intake, and 2 green bars representing resulting biomarker improvements, reductions in body weight and PPG. It visually reinforces the conclusion that lifestyle efforts are significantly greater than the corresponding biomarker outcomes. These visualizations further support the finding that it requires 3 to 4 times of lifestyle effort to achieve 1 time of biomarker improvement.